All right, now I can safely assume that you guys are experts on DNA structure. So now we're going to go into the next section, how DNA replicates itself or makes copies of itself. Normally this is going to happen just before mitosis in interphase, in the S phase of interphase. We said that you have to have 2 copies of all of the chromatin. Well, this is what's happening here. Since the chromatin is made up mostly of DNA, and that's all the important stuff, this is how it gets copied. Now, by the time we're done here, you should be able to sketch and describe the process of DNA replication, meaning you should go ahead and from scratch, from just like a blank piece of paper, go ahead and sketch everything out and describe what's happening. There's a couple of important enzymes, helicase and polymerase. You'll have to be able to describe their function, and you'll be able to hopefully define what's meant by semi-conservative replication. All right, so DNA replication is the process by which all these DNA molecules can make duplicate copies of themselves. So we're going to start out with a parent strand of DNA, our original strand of DNA. And rather than making a full ladder diagram, I'm just going to write our little uprights here and assume that you already know that this is alternating sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And we can put the nitrogen bases over here. We'll put thymine, adenine, cytosine, another adenine, thymine, guanine, and let's leave off a cytosine here. And if that's the left half, then you can figure out what the right half should be. So in your mind's eye, think of sugar, phosphate, sugar, phosphate, sugar, phosphate. And adenine always matches up with thymine. Thymine with adenine. Guanine with cytosine. Thymine with adenine. Adenine with thymine. Cytosine with guanine. And guanine with cytosine. And so on all the way down throughout its length. Well, the first thing that's going to happen is a special enzyme called helicase, DNA helicase, is going to actually cause the hydrogen bonds that are between these nitrogen bases down the center, the rungs of the ladder, all the way down the center, to break apart, causing DNA to kind of sort of unzip down the middle. Imagine this ladder, or this half of the ladder, and this half of the ladder as just being separated. So imagine our parent DNA split apart now by the helicase, exposing both the left and the right sides. Cytosine is still going to want to attract a guanine. Thymine is still going to attract an adenine. And we've got all of these potential free nucleotides down here which act as kind of spare parts. So imagine one of these spare parts lining up alongside its counterpart. Guanine next to cytosine, cytosine next to guanine, adenine next to thymine, and the same thing happening on the other side. Meanwhile, other free nucleotides would float up, eventually find their place, and the left half serves as a template or a mold for a new right half. And the old right half serves as a template for the old left half. What you eventually end up with is, of course, 2 new strands, each of them identical to each other, and each identical to the original parent strand where you started from. Now the cool thing is, even though these things split down the middle, the entire left half and the entire right half are still conserved or preserved. Therefore, the sequence of nucleotides from top to bottom are the same. That means that nothing has changed, which is really cool because, hey, that's the genetic code. You don't want that to change. If it changes, that's a mutation. Now, since the original left half of the parent strand and right half of the parent strand still exist, this is called semi-conservative replication. Each half of the new strands, or each new strand, has half of the original DNA. Kind of cool. In this way, the genetic code can be copied, but also preserved generation to generation. Now in a test, exam, quiz, whatever, you'd be responsible for sketching and describing steps 1, 2, and 3, which we just went over, and are summarized again for you right here. You'll need to do this on the exam as one of the short answer questions. So yeah, be prepared to draw a sketch. But if you want some extra credit, we can show you the rest of the story. Turns out that if all you really want to do is understand that DNA replicates itself, what we've done is fine. But it's a little bit more complicated. Turns out that we have to worry about how DNA copies itself in different directions. For that, we need to look at the DNA molecule again. Okay, so here's a kind of a stylized drawing of a DNA molecule. It's not a perfect double helix, but we can still see this untwisted helix as our ladder, with our sugars and phosphates going on either side. And we said, yeah, these things are kind of sort of parallel to each other, but if you look at the direction that the sugars are pointing over here on the left side, notice that they're pointing in a different direction down on the right side. So they call this arrangement being not so much parallel but anti-parallel, because they're facing in different directions. Now, this is important because, as it turns out, DNA can only copy itself in one direction at a time. So sometimes they worry about DNA molecules and the way they're facing, and they will label these as the so-called 5' end and 3' end. Why 5 and 3? Well, it has to do with the way they kind of sort of number the carbon atoms that make up these inner rings. And the first carbon is over here, second carbon, third carbon, so the third carbon is facing downhill here. Fourth carbon, and the fifth carbon is attached to this phosphate over here facing uphill. Over here you have the opposite sequence, so the 5' end is facing downhill, and the 3' end is facing upwards. Now, again, directionality is going to be important because DNA can only copy itself in one direction. So here we go for some exercise. So here's the rule. RNA polymerase only works in one direction. So the DNA can only be copied in a 5 to 3 direction. So one new strand is copied continuously from that 3 to 5 template, the 3 to 5 mold. Replication of the other strand has to go in the opposite direction. Now normally that would mean that the entire DNA molecule would have to separate all at once, and this just doesn't happen because the molecule is much too long and it's sticky, it's going to get all gummed up, and it is just not going to work. So instead of unzipping completely down its entire length, DNA will begin to unzip at specific sites known as origins of replication. You would see one right here. Okay? The rest of the molecule is still more or less intact, but it's unzipped at this point. So DNA replication can occur here at this location. Now imagine new nucleotides lining up here. And we would continue in this direction. But instead of starting down here, we'd have to start up here. And new nucleotides would be started up here, and we would continue in this direction. Thus, the so-called leading strand is going to be synthesized and created continuously from the 5' to the 3', or at least the new 5' to 3', right, using the 3 to 5 as an old template, and we'll go continuously in this direction. No problem. But the lagging strand, the so-called lagging strand, has to go in the opposite direction and has to be copied in a series of short fragments. So sure, we can start at this replicating fork and get to the end, but we have to go ahead and then start a new replicating fork so we can get another fragment done. So if we left off here, we can just go ahead and keep on going. As long as that replication fork gets pushed forward, we can just synthesize continuously along this direction. However, if we left off here going in this direction, we have to go ahead and start over here again and now try and connect them. These little short fragments here would be known as Okazaki fragments. Later we'd have to kind of sort of splice these together so that we could have a complete fragment of DNA. Yeah, kind of a cleaned up version. It's a lot better than my drawings. But here's that leading strand synthesized continuously in this direction. And here's the results of those Okazaki fragments. Our first replication fork was over here somewhere, so that's our first fragment. And then we had our second fragment, and we had our third fragment. We're going to start our fourth fragment over here. And eventually what we'll do is we'll splice all these together. Don't worry about these other enzymes, primers, ligases. We'll save that for an additional biology class. But directionality kind of sort of plays a role here. So if you wanted to explain directionality in terms of DNA replication, then yeah, extra credit for you. Now usually there's supposed to be a practice quiz at the end of these. And hey, no quiz, but you do have to go ahead and have a little bit of a project. So get a clean white piece of paper and provide a series of sketches. And they should be similar to the simple ladder diagrams that we showed in the video here. And you want to document all the events of DNA replication. Helicase splitting apart the parent DNA, free-floating nucleotides joining to either the left or the right side, and eventually ending up with two new identical pieces of DNA. You don't necessarily have to go into the directionality aspect and explain Okazaki fragments. If you want to, you can do it for extra credit, but you should at least get the simple version. Okay? Important because you're going to be asked to duplicate that series of sketches on the next exam. So, yeah, good luck, and this will be due next time I see you. Have fun with it. Later, bio buddies.